Anybody get to do anything fun?
Amazing.
How many of you went home? I hope you didn't bring anything back. I hope you didn't take anything home. I know a lot of schools actually beg their students not to go home. I think UK did, so now my son. What?
That's hard if you live like 5 minutes down the street.
Yeah, oh, I know.
I do. And I'm just like, I gotta go home and see my dog. But
It's a tough time.
Yeah.
There's no
maybe
No
that's
doubt about it. Any of you have younger siblings who are having to deal with virtual school or, I'm really glad I don't have little kids. I mean, I think the high school students can probably cope with it. It's not fun.
the high school are kind of doing it by themselves though.
What?
I said the high school students are kind of like doing it by themselves though because they showed us how to do it.
I don't know. I would hope so.
Yeah, well it's kind of like MTI. I know a few people who are doing it.
Yeah,
It's kind of like self-explanatory for the high school students.
yeah. But little kids, I mean, trying to teach kindergarten online, I'm like, not sure how you do that. But I guess you figure it out. Hopefully kids will be able to go back, it's Kentucky, September 28th, isn't it?
Oh, I thought it was October.
Now, right now, well, it depends on the school system,
Oh.
but the governor has said, has asked schools not to go back until September 28th, I think. So anyway. Okay, so this is where we were on Friday, and I told you I'd tell you a story about, somebody asked about the blood test. Okay, so when I was pregnant with Daniel, so that would have been 1989, or 1988, he was born in March. The doctor offered me this blood test, wasn't required, it was called the alpha-fetoprotein test. And I was like, okay, what's that? And so she gave me the little brochure and said, if you want to take it, we'll do it next visit. So I read the little brochure. And what the alpha-fetoprotein test is, it's a test on the mother's blood that will have a certain level of what's called alpha-fetoprotein. Okay, it's just a protein that's found in the fetus. And if the level of alpha-fetoprotein is high in the mother, it could mean that the baby has what we call a neural tube defect. Okay, there's two major kinds of neural tube defects. First is anencephaly. Anencephaly means basically no brain. Okay, that's one of those incredibly sad, tragic events. Babies who are anencephalic, it's not that they have developmental delays. They don't have a brain. They have a brain stem, so they can breathe and have a heartbeat, at least for a while. But they don't have any of the other structures that are necessary for what we consider to be human life, okay? There's no thinking, there's no emotion, there's no any of those kind of things in the brain. Yes?
Is that the one where like the babies are born and their eye sockets are extremely large?
I'm not sure.
It's kind of like protruding out of their head.
It could be, I mean, the skull is like,
Their skull is
caved in. It's like the whole back, the whole part of your head that would normally house your brain isn't there. These babies generally, without massive support, will die very quickly. Most people will terminate a pregnancy because it's not viable, basically. A few people will continue a pregnancy because they believe that. Some people will continue the pregnancy because they want to be able to donate the organs, because there aren't very many newborn organs available if somebody needed a heart or kidney or something. Okay, that's everybody's choice. The second type of neural tube defect is called spinal bifida, and you may very well know somebody who has spinal bifida. It can range, basically it means that the spinal cord is exposed. it depends where and how severe it is as to what the outcome is. It can range anywhere from really severe physical and mental problems, mental meaning developmental, mental retardation, that kind of stuff, all the way to basically having no impact at all. There's a young man in our church, well, he's not so young anymore, he's probably 45 or 50, but who has spinal bifida, and he used a wheelchair. He has recently gotten a chair, the electric chair. For a long, long time, he had a regular wheelchair. He did not basically have feeling from about the waist down. Good news, bad news is his family is real prone to kidney stones, and so is he, but he never feels the severe pain from kidney stones. But he doesn't necessarily know he has one. So that's kind of a good thing, bad thing. So anyway, so that's spinal bifida. So what I asked my physician was, if the baby is anencephalic, I would probably terminate, okay? Will that show up on an ultrasound? The answer is yes, undoubtedly. If the baby has spinal bifida, probably wouldn't terminate. But the advantage of knowing at that point is that you would depending on the circumstances, you would probably want to have a cesarean, because you would not want a damaged spinal cord going through the birth process. That could be further damage the spinal cord. So I said, will that show up on an ultrasound? And she said, almost always. I said, okay. So I don't need to test for those two things. If the amount of alpha-fetoprotein is low, that can mean that the baby has Down syndrome or another one of the trisomies. Generally, it's Down syndrome. And I said, I would not terminate for Down syndrome. So, and when I read the literature, 90% were false positives. What that means is 90% of the time that it's either high or that it's low, there's nothing wrong. 90% of the time. Well, if it came back low, which is typical, which is more common than coming back high, I would not have the amniocentesis, because I wouldn't terminate anyway, and the amniocentesis carries a risk of a miscarriage. And so I said, why would I take this test? Because if I'm not going to have the amnio, all I would do if it came back low is spend my entire pregnancy worrying and freaked out about it. Okay? So I said no to the test. I just won't take it. Fine. Find out later that a friend of mine took the test. Now, Diane was like the most pro-life person you can imagine. She was, or she still is, really, really big on God's will, and everything's God's will, and everything that happens is God's will, and all this kind of stuff. And but she took the test. She's educated. Her husband's a pharmacist. They were educated people. And she took the alpha-fetoprotein test, and it came back low. And then she had the amniocentesis. Okay, now let me tell you something else about Diane. She had a hard time getting pregnant. She had endometriosis. Endometriosis is where the endometrial tissue, which is what a woman sheds every month in her period, where that tissue grows outside the uterus. It grows somewhere in the abdominal cavity. It can cause really severe pain. And she never had that. But nowadays, see, we're talking 35 years ago. Well, my son's 31, so over 30 years ago, because her daughter's like 33 or 34. Nowadays, they just zap it with laser surgery, okay? I don't mean to minimize it, but it's relatively easy to treat. But back then, they didn't have laser surgery. And the way they treated it was to give a woman male hormones. Basically, they gave her testosterone, which would then suppress the growth of that endometrial tissue. And you did it for a year. So this meant Diane didn't have periods for a year, it meant that she had all the side effects of a woman taking testosterone. Her voice deepened. She grew a little bit of facial hair. She probably had acne. You know, all those kinds of things that happen when you take testosterone. And she's a musician. I mentioned that. She's a musician. Her voice change is permanent because it makes the vocal cords bigger. It does something to the vocal cords. And that doesn't go away. So my point is she worked really hard to have this baby. And then she not only took the alpha-fetoprotein test, and she would never terminate for any reason, she also had the amniocentesis. I'm like, Diane, you have got to be kidding me. Why would you do that and risk a miscarriage after you worked so hard to get pregnant, and then have this test when you wouldn't terminate anyway? What difference does it make? And her response was, we wanted to know ahead of time so that we could grieve ahead of time and be prepared when the baby was born. I'm like, you're nuts. That's all I can say. You are absolutely nuts. Now, who made the right decision? By the way, her baby was fine. Who made the right decision? I think I made the right decision. She thinks she made the right decision. Why do I tell you this long, boring story about people you don't know? Because you're going to face these kinds of decisions. Just because a test is available does not mean you have to take it. Okay? For me, it was a no-brainer. I don't want to. If I wouldn't do anything with the information, why would I want it? If I could do something with the information, then I want the information. Fast forward several years, and David went to this preschool called Push in Frankfurt that it's not there anymore, but it was a reverse, what do they call it? Reverse mainstream, where they served about half the students had some type of handicap, needed therapy, physical therapy, occupational therapy, speech therapy, something. And the other half of the kids were typical, what they consider typical kids. So he went to this preschool. And this little girl, I think she was a year younger than him, was in the preschool, and I got to, not friends with her mom, but certainly acquaintances. And I saw her one day, and this little child had some of the features of Down syndrome. Her face looked a little bit like a child who has Down syndrome. Her speech was a little bit like somebody who had, she was there for speech therapy, so with David for the first year. So, you know, she had a few of the little signs of Down syndrome, but she didn't have any intellectual disability. So her mom saw me one time, she said, I want to ask you something. She said, the school wants me to have her tested to see if she has a little piece of extra chromosome 21 because she has certain features. And I said, does she have any trouble in school? No. Behavioral, emotional, educational, any issues? And she said no. And I'm like, then why would you want her labeled? Why would you want to have a test done that would label her with something if there's not a problem? I said, I wouldn't do it. Now, before she has children, she might want to know. I said, that might be a useful piece of information. But when she's 10, no, it doesn't matter. So I'm a person who says more information is not necessarily better. Many of my friends want all the information available. Now, what they do today with the alpha-fetoprotein test is a little bit different than what they did 30 years ago. Now they combine the alpha-fetoprotein test with a measurement of the lengths of the long bones, particularly the arms, because there is a correlation if the alpha-fetoprotein is this much low and the arm length is so much, the comparison between the humerus and the ulna or something, they do a ratio. If it meets a certain criteria, then there's a more likelihood that the child has Down syndrome. So more people are taking the test now. But again, the child I told you about that was conceived through where they actually inject the sperm into the egg, her alpha fetoprotein came back low, and the first time they did the measurements, it fit the criteria for Down syndrome. And they really wanted her to have the amniocentesis. She was kind of like me, I'm not going to terminate anyway, so I'm not going to do it. And then when they had the next couple of rounds of measuring and stuff, it was fine, and the child was fine. So, you know, would I have the test today when you could add in those other parts? I don't know if I would still do it, because again, I would just obsess. I'm the kind of person who would obsess over it. So, I tend to not do things if it's going to make me obsess over it. So, that's my story on having the test. And again, all of you have to make your own decision. Any questions about it? Okay, that's the end of chapter 9. Okay. Now chapter 10. Okay, in chapter 10, what we're going to do, hopefully, is figure out why having this gene on this chromosome makes you have brown hair or blue eyes or whatever. We're going to look at the actual, how does a gene turn into a trait? Okay. So that's the molecular biology of the gene. First, we're going to talk about DNA replication, then we're going to talk about what's called central dogma. Okay, so DNA replication. They knew about DNA. Remember, chemistry is ahead of biology, physics is ahead of chemistry, math's ahead of physics. Okay, so chemistry knew by 1900 that there was DNA. They had already isolated DNA. They did not know at that time it was the genetic material because they knew chromosomes were DNA and protein. They weren't sure which one was which. But they finally figured out that DNA has the capacity to store genetic information, and that information can then be passed on from generation to generation, whether you're talking about cells or you're talking about whole multicellular organisms. So remember that DNA and RNA are nucleic acids. They are made up of nucleotides. The nucleotides are joined with that sugar phosphate backbone. So if we look at the structure here, this is a polynucleotide. Poly means many. Nucleotide means the building block units of DNA. So an individual nucleotide, let's look at it again, is kind of complex. You have what we call a nitrogenous base, and that just means a ring structure that has nitrogen in it. That way. A nitrogenous base, which can be the A, C, T, or G, adenine, guanine, cytosine, thymine. Attached to that base is a sugar. If it's DNA, it's a deoxyribose sugar. If it's RNA, it's a ribose sugar. And that has to do whether this carbon has any oxygens attached to it. I wouldn't expect you to pick that out at this point, but that's what it means. Okay? Deoxy sugar is missing one of the oxygens it should have. So you got this 5-membered ring, and then you have a phosphate group. If you can remember that ATP is basically a nucleotide, it's actually an RNA nucleotide because it's a ribose sugar, not a deoxyribose sugar. If you can remember that ATP is a nucleotide, that will help you because that will help you remember it has a phosphate group, okay? So we got a base, a sugar, and a phosphate. The rail, what we call the rail of the ladder, are these alternating sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate units. And those are put together through dehydration synthesis. What does dehydration synthesis mean?
You break it apart.
You're putting something together by removing water, right? Okay? So synthesis, you put things together. Dehydration, removing water, okay? So these sugar phosphate units are hooked together with dehydration synthesis. This is what it would look like in a simplified version. This is a little more detail of what it looks like. The four nucleotides found in DNA differ in their bases, A, C, T, and G. And remember that uracil has a U instead of a T. If you see a string of nucleotides and there is a T in there, you know it's going to be DNA. If you see a string of nucleotides and there's a U, then you know it's RNA. Okay? Now, Watson and Crick won the Nobel Prize for discovering the double helix structure of DNA. They stole the work of Rosalind Franklin. My son and his fiance now have a little kitty cat that they named Rosalind after Rosalind Franklin. But Watson and Crick stole her X-ray crystallography data which gave them the information that it was indeed a double helix, and never gave her credit for her assistance in figuring out the structure of DNA. Crick has died. I'm not sure if Watson's still living or not. Last time I looked him up, he was still living. He's very old. But Watson is a really creepy guy, okay? He's actually been kicked out at least once and maybe twice out of the Royal Academy of Sciences, and he's British. He espoused not only eugenics. You guys familiar with the concept of eugenics? Kind of part of Hitler's thing, that there are, you can make designer people, that we should stop people from breeding if they have conditions that we think are undesirable, or we should breed people to be workers or basically slaves versus people who are more intellectual and that kind of stuff. And a lot of people bought into that in the early 20th century, unfortunately. But he was a big espouser of that. He was a racist, still is a racist, and has never recanted any of that, so he was eventually kicked out of the Royal Academy of Sciences because he's a very bad person. Maybe a good scientist, but he's a bad person. But they are the ones who figured out the structure of DNA with Rosalind Franklin's help. So we typically look at DNA as if it was a ladder. It's just an easy image that most of us are familiar with, looking at a ladder. And if you look at that ladder with twist, particular kind of twist, that's what DNA looks like. We often untwist it so that we can see it more simply. Okay? So again, along the rails here, going up and down, are the sugar phosphate units. Going across, the rungs of the ladder, which you put your feet on, those are the bases, the base pairs of the DNA. So there's different ways of representing it. This is called the ribbon model. This is a more detailed model, and this actually has the atoms in it. If you look at this model right here, you can see that the hydrogen bonds, there's 2 hydrogen bonds holding A and T together, and there's 3 hydrogen bonds holding C and G together. I think that's kind of interesting. Okay? So it means it's going to be easier to break apart A's and T's than it is C's and G's. Also notice that A's and G's, you probably can't see it real well from here, but this is an A, this is an adenine, this is a guanine, have a double ring structure. They are called purines. The T and the C have a single ring structure, and those are called pyrimidines. Okay. We always pair purine with a pyrimidine, and that gives, one of the things they were able to measure fairly early was the distance between, how wide that double helix was. And it doesn't work if you put two pyrimidines together or two purines together. But it does work if you put a purine with a pyrimidine. Okay, so that's how they kind of match up. So the rungs of the ladders are actually these structures right here. Does that make sense? Whereas the rails are these sugar phosphates. And that's where the dehydration synthesis takes place. Okay. The reason A meets with T and C meets with G just has to do with where those hydrogen bonds can form. The hydrogen bonds won't form between A and C. They're just not in the right position. So how does DNA replicate? Before a cell can divide, remember in interphase, the DNA has to replicate. It has to do so with tremendous precision. We know that a mistake is made about 1 in a million times. That means almost all the time, 999,999 times out of a million, it's done properly. So the mechanism of replicating DNA, will go into more detail next semester. But what happens is, if this is the parental DNA molecule here in blue, what happens is it basically unzips. The best image I can give you is that it unzips, and each side then becomes a template for a new piece of DNA. And then you have a daughter cell that is identical to the parent cell, the parent DNA. You have the daughter DNA that's identical to the parent DNA, barring an error. There are environmental things that increase the damage or the mutation rate, and one of those is UV light. UV light increases the rate of mutations, particularly in skin cells, and that's why it can lead to skin cancer. Anytime you get a tan, that is your body's attempt to basically block UV light. When you get a sunburn, you have damaged your skin. Sunburn is a bad thing. It is not good for your skin. The more sunburns a child has before the age of 18, the more likely that child is to have skin cancer as an adult. You all are mostly past age 18. That doesn't give you a free pass to get lots of UV light now, but most of you will be parents someday. I was the mom who ran across the soccer field smearing sunscreen on any child I could get my hands on. Today I'd probably get sued by the parents or something. But I was really very, very careful. I told my children they'd appreciate it someday. Okay. So how does this happen? In eukaryotic cells, we get multiple origins of replication, and then the DNA replication proceeds in both directions until they meet, and then you have completely copied your DNA. Let's see if this video will work.
molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made.
So do you get the idea? You have to go past it and make it toward the replication fork. And then you go past it, make it toward the replication fork. Whereas the other direction, you can just keep making it as it opens up. If anybody sews, I think of it as a back stitch, where you go back and pick it up.
And the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, how do we go back to the... So, they mentioned several different enzymes that are involved. Do you remember the first enzyme? Helicase. Okay, I'm not spelling these right. Okay. Then primase. Then DNA polymerase 3. Okay, what was next? I think they mentioned one we don't usually worry about. What?
DNA library.
Well, yeah, we're gonna do DNA, gotta have DNA polymerase 1. There was one in between there that actually removed I think it was a nuclease that removed the RNA nucleotides. We don't usually worry about that one. But DNA polymerase 1 is what fills in the RNA, where the RNA were, and then ligase. Which hooks everything together at the end.
What does the second and third one do again?
Trying to see if I can get this on here. It's not very in focus. Say that again.
What does the second one and third one mean again? The primase and DNA colon?
Oh.
The third one?
The primase puts on that should be AFC, not IFC. The primase puts on the RNA. Because you can't start with DNA. There's several reasons they think that RNA is more ancient than DNA, and this is one of those reasons. Because you cannot start the replicate, you cannot start a strand with DNA. You have to start with RNA. So that's what the primase does. Then the DNA polymerase 3 attaches then to that short strand of RNA. It attaches DNA nucleotides. And then I think it was what did I say it was? Another enzyme a nuclease that comes and takes off the RNA bases, and then DNA polymerase 1 comes along and puts in the correct DNA. And then the ligase hooks all the ends together. Okay. There's also a video on your Mastering Biology that kind of goes through this process. But what you end up with, the big take-home message here, is that you end up with 2 strands of new DNA. Went from one original DNA to 2 daughter DNA molecules, and each one of those daughter DNA molecules has one strand that's the parent from the parent, and one strand that's brand new. And we think that was favored because you have less errors that way. Okay? So that's all we're going to talk about for DNA replication. Again, we'll do a lot more detail in 212. The flow of genetic information from DNA to RNA to protein is something that is referred to by some people as central dogma. What is a dogma? What does the word dogma mean? There's certain things in religion that are kind of considered dogmas. Kind of what's the bottom line thing? If your Christianity is the one I'm most familiar with, what's kind of the bottom line belief in Christianity as opposed to other monotheistic like Judaism, Islam? Jesus is the Messiah. Sort of. What characterizes Jesus? I'm sorry? He's the Son of God. He's also what? The Son of Mary. Jesus is fully God, fully human. That's a Christian dogma, that Jesus is both fully God and fully human. Okay? So so we're talking like really, really, really important. And the flow of information from DNA to RNA to protein is then sometimes called the central dogma of biology. So DNA functions as the inherited directions for a cell or an organism. So how do we do this? How do we get from the gene to the fact that your eyes have a color? From your gene to how much insulin you make? Okay, all those kinds of things. So our genetic makeup is the sequence of nucleotides, which then leads to certain traits. So here's kind of the bottom line. We have something called transcription and translation. I usually write it this way. This step is called transcription. This step is called translation. Okay? DNA to RNA to protein. DNA to RNA is transcription. DNA or RNA to protein is translation. Now, let me tell you, when I was sitting in your seat, I have a really hard time remembering which one is transcription and which one is translation. The words sound way too much alike. And now I've figured it out. I mean, eventually, figure things out. But I have to tell you, the way I remembered it when I was first learning it is finally, since I kept getting confused, is transcription comes first alphabetically compared to translation. That is literally how I finally learned it. Okay? But now let's talk about a better way to learn it. I mean, I'm for whatever works, but think about the word transcription. Have you ever heard of a transcript?
Yes.
Where do we hear of a transcript?
Doctors write transcripts.
Doctors write transcripts. Courts have transcripts of court proceedings. In the olden days, you could send off for a transcript of a talk show. Nowadays, we just video stuff, but
Yeah.
back then, you could actually get a transcript of something. If the doctor speaks in English, what language is the transcript in?
English.
English. If the doctor speaks in Spanish, what language is the transcript in? Spanish. Okay? So transcription, you're using the same language. Okay? And going from DNA to RNA, you're using the language of nucleic acids. Okay? Now, the word translation. What does translation mean? Translation means changing languages. Okay? Going from Spanish to English, or English to German, or German to Japanese or something. Okay? So translation means you're changing the language. And when you go from RNA, which is a nucleic acid, to protein, which is a protein, you're changing languages. You're going to a completely different language. Now, let me caution you to understand that this is something people sometimes don't get. We don't turn DNA into RNA. We don't turn RNA into protein. DNA serves as a template or a model to make RNA, and RNA serves as a template to make protein. But we don't turn one into the other. Okay? The original DNA still exists, the original RNA still exists. All right. So, this is just saying kind of the same thing. The one gene, one protein hypothesis states that the function of an individual gene is to dictate the production of a specific protein. They've gone through several iterations of this that are way too detailed to get into right now, but it used to be one gene, one enzyme, then it was one gene, one protein. Now they usually say one gene, one polypeptide. So those are kind of subtle differences that aren't necessarily relevant for us right now. So here's a kind of an overview of how it happens. So we have a DNA strand that has a particular code. When it is transcribed, we make RNA that matches up to that code. But remember, RNA is going to contain uracil, not thymine. And then every 3 RNA pairs we call a codon. And each 3 of these codons, each 3 of these bases, which make up a codon, code for one and only one amino acid. So that's how it's going to work. Okay, so transcription. Genetic information travels, is transferred from DNA to RNA. So the DNA serves as a template. So how does that happen? Okay, in this picture, the blue is the DNA and the red is the RNA. And your textbook is pretty consistent using the same color to mean the same thing in different pictures. So here's our DNA coming along here. And you'll notice on both sides, the DNA is in its double helix. But the part that is actually being copied will be open. It's like having almost a broken zipper, okay? You have a zipper that will open in the middle, okay? And then zipper back up. So that's what's happening here. So this part is getting ready to zipper back up. This part is unzipping. So the direction is going from left to right. So the RNA polymerase attaches at a section of the DNA we call a promoter. In this picture, it's tinted green. It's a little hard to see. The RNA polymerase attaches. It starts the RNA in a process we call initiation. It then goes through a process we call elongation and makes a longer transcript. And then it'll hit the stop and it will terminate. So the 3 steps are initiation, elongation, termination. And those words are pretty self-explanatory. Initiation, you're starting. Elongation, you're making it longer. Termination, you're stopping it. Then you have what we call the primary transcript. The main gene we're going to the main enzyme is RNA polymerase. So we use DNA polymerase to copy DNA to make more DNA. We use RNA polymerase to make RNA. To initiate it, there's actually a signal that says, hey, come here, it's me, start. Okay? And the RNA attaches then to that promoter, which is yelling, hey, come and get me. And there's all kinds of factors, and again, we'll go into a lot more detail in 212, but all cells aren't making the same proteins at the same time. So every cell has reasons why it makes a particular protein. So when you start thinking like a scientist, every time we figure something out, you got to figure out what came before that. Okay? So somehow the cell signals and figures out that, hey, this is a protein we need to make right now. And so it recruits what it needs to get things going. And that's part of the initiation. So the initiation is really quite complex. There's a lot that goes on to this initiation. And then the RNA polymerase starts. The elongation just means it's getting longer and longer. Termination just means it reaches the stop and kind of falls all apart. Okay. Now, this was, of course, first studied in bacteria. We study almost everything in bacteria first. Bacteria are prokaryotic cells, right? Most other organisms are eukaryotic cells. So we studied in prokaryotic cells. So pretty much in prokaryotic cells, once you made your RNA, you're ready to make a protein. Eukaryotic cells, they were kind of shocked to find out it doesn't work that way. Because what a eukaryotic cell does is process the RNA after transcription. And there's a couple of steps to this. Okay? So, one of the steps is adding a cap and a tail. Okay? We actually add a GTP on the front, and we add what's called a poly-A tail, a whole bunch of adenines on the end. Turns out one of the reasons, remember I said RNA probably came first, one of the reasons DNA probably came into existence is because RNA is easily degraded. DNA is tougher, so to speak. So eukaryotic cells modify RNA to try and make it live longer, basically. So adding this cap and tail, that poly-A tail, helps retain the rest of the RNA because enzyme will come and cut off the tail first. So that's one thing that happens. This cap is attached, the tail is attached. And then something called introns are removed. Now, this is a case where, again, if I was in charge of the world, I would change the names of things. If you heard the name introns and exons, which one would you think you kept? Introns. And which would you get rid of? Exons. That sounds reasonable. It's the exact opposite. Intron stands for intervening segments or something like that. And exons stand for the parts that are expressed. So it's confusing. So this is one of those cases where the words kind of mean the opposite of what you might first think. Okay? So introns are removed, and you actually end up with a coding sequence or a mature RNA. Sometimes this is called immature RNA, sometimes it's called the primary transcript. But in eukaryotic cells, it's largely altered before it actually leaves the nucleus and codes for making the proteins. It's usually much shorter, and in fact, sometimes it's a third as long as it started out. So, you all familiar with the VCR tapes?
Yeah.
Okay. Have you ever seen old movie reels?
Yeah.
Where it's literally tape. There's a kind of tape, the movie or the music or whatever is on. Well, when I was in college, my boyfriend was the one who made the mixtapes for the fraternity parties, okay? And that was his job because he had a reel-to-reel tape player, which was a big deal back then. And the bigger the tape, the better the sound quality. So he had this nice big contraption. And he would literally find the song, take a pair of scissors and snip it, and then you would attach it to the end of the last song with a special kind of Scotch tape, and you could get a different mix for every party. You could put it together differently for every party. We do splicing now, mostly electronically. My husband likes to have a separate ringtone for everybody, okay? And so he takes snippets of music and turns it into a ringtone. And so he splices music electronically. I don't know how to do that. Everybody has the same ringtone on my phone. Okay. I'm Brown Eyed Girl on his phone. Okay. So that's what splicing means. You cut out parts and then attach the ends back together. When you move from transcription to translation, we are converting again from the language of nucleic acids to the language of protein. Here we use we're going to talk about 3 kinds of RNA. Next semester you'll get 3 more kinds of RNA. So we're going to talk about messenger RNA, ribosomal RNA, and transfer RNA. And these are abbreviated mRNA, rRNA, tRNA. Okay. All right, so messenger RNA is what we think of as the RNA. It is the transcript. It is what comes out of the nucleus ready to be the template for the protein. All RNA is a product of transcription, and it is the part that moves from the nucleus to the cytoplasm. And it is what contains the codons. So the big deal about the messenger RNA is it contains the codon. So what is a codon? A codon is a triplet of bases. If we think about it mathematically, how many amino acids do we have to be able to code for? You guys remember? How many important amino acids are there? There's 20 amino acids. There are 4 bases. So from a strictly mathematical perspective, if you take 4 bases, one at a time, in any order, how many things can you code for? You can only code for 4 things. A stands for something, C stands for something, T stands for something, G stands for something. That's it. Now, what if you took 4 codes, 2 at a time, in any order? How many codes would you get? You could do AA, AC, AG, AT, right? Then you could do CA, CC, CG, CT. Get the pattern?
Yeah.
Then you could do GA, GC, GG, GT. So how many total codes do you get? You get 16. Okay? Because the last row would be then, oh, this is supposed to be RNA, sorry. Everywhere there, I wrote a T, there should be a U. UA, UC, UG, UU. Okay? So you get 16 codes. But how many amino acids do we have? 20. So that's not enough. So it turns out we have to have triplets, which gives us a total of 64 codes. So here's the first one, the second one, and then the third one for each set. So this is the genetic code. UUU, UUC, UUA, UUG. And just go on what we say about the genetic code is that it's specific. Each codon codes for one and only one amino acid, but is also redundant. And that means that more than one code can code for the same amino acid. And in fact, if you look at leucine, for example, there's actually 6 different codes for leucine. Most of them have 4. Most of the amino acids have 4 codes. And that third base, we sometimes call that third base kind of a wobble. Because in this case, it doesn't matter what the third base is. Proline, if you have CC, it doesn't really matter what the third base is. It's going to be proline. And so we call that wobble. It doesn't really matter. So an awful lot of them have 4. The rest mostly have 2. And then we have a couple, I think there's 2 that have 6, 5 or 6. And then 3 of the codes are stop codes. And then there's a couple, methionine and tryptophan, that only have 1. Okay? So that's our genetic code. Now, usually I tell the class you have to memorize this, get started. But y'all are under enough stress, so I'm not going to tell you that and then teach you.
How many kids were there?
There's 64. Nobody has the genetic code memorized unless they use it. To me, it's like chemistry professors who want you to memorize the periodic table. Pretty readily available. The only people who really have it, who really know it, are the people who use it. When I was teaching chemistry, I could tell you the atomic weight of almost any element. I can't do that anymore. I can do the most common ones because I don't use it all the time. Okay? So it's one of those things. If you use it, you know it. If you're a biochemist doing nucleic acids, you're going to know it. Just pure experience with it. But no, you don't have to memorize genetic code. It's readily available. And you can pull it up on your phone for that matter. On the exam, I will provide you with a genetic code. Okay? So that's the messenger RNA. The transfer RNA does kind of what it sounds like. It transfers an amino acid from point A to point B. In space, it kind of looks like this. That's a 2-dimensional, it's actually a 3-dimensional structure. But looks something like this. One end is the amino acid attachment site. An opposite end is where the anticodon is. The anticodon is what matches up to the codon. So if the codon is CCC, what's the anticodon going to be? CCC will match with GGG. Okay? So those will match together. So the anticodon is the complement of the codon. So that's on one end, and then the attachment for the amino acid is on a different side, different part. So technically, the transfer RNA basically acts as a molecular interpreter. And it carries the amino acids basically from loose in the cytoplasm to the ribosome. Ribosomes, which are made up of ribosomal RNA and protein, are the site. They provide maybe the assembly line would be an analogy. They provide the actual site for the making of the protein. Ribosomes have 2 subunits. They have a small subunit and a large subunit. And they have, actually there's 3 sites, but here there's 2 sites, an A site and a P site. It stands for, the A site is the first one, the P is the second. Just go with that for right now. P is you're adding to the peptide, where you make the peptide bond. A is the starting point. So what happens is you have to have all 3 of these RNAs your transfer RNA, and here is your ribosomal RNA, blue. So the blue is the ribosome, the red is the messenger RNA, and the green here is the transfer RNA. So you gotta have all three of them to get things going. So what's the process? Again, we have initiation, elongation, termination, same three steps. And again, initiation is the most complex of the three. So the first phase, you have to bring together all the stuff you need. The messenger RNA, the first amino acid with the transfer RNA, and the two subunits of the ribosome. So that's how stuff starts. So here is the messenger RNA, and it's what contains the codons. There's the cap, there's the tail. Here's where the message actually starts, where the codons actually start. To initiate, what you have to do, you always start with AUG. The first codon in eukaryotic cells is always AUG. That's the only one I know. I know that AUG codes for methionine, because that's always the first amino acid in eukaryotic cells. So the small subunit of the ribosome, the messenger RNA, and that first transfer RNA that contains methionine all have to come together. Until that happens, nothing happens. That's how you start it. As soon as these three things are together, the large subunit will come in and attach itself. And then this transfer RNA is actually sitting in the P site. So that's how the process gets started. Elongation means that transfer RNAs keep coming in. Is there a bug? Is it a dead bug now?
No, I was just stuck on it, disappeared somewhere.
Oh, that's always fun. I love that when I'm going to bed and you miss. You're like, okay. All right. So the anticodon has to find its correct amino acid, the anticodon, the transfer RNA that has the anticodon has to find the right amino acid. And then that anticodon binds to the codon, a peptide bond forms, and then it translocates. So this is kind of what it looks like. So here's our growing polypeptide. This is the P site, this is the A site. So we have the codon sitting here waiting for the anticodon. The anticodon of the transfer RNA, which has its amino acid, comes and sits in the A site. And while it's sitting there, a new bond forms. This is a peptide bond that's forming. It forms between this amino acid right here and this incoming amino acid. And when that peptide bond forms, this bond breaks. So the bond here is going to break. Then that transfer RNA leaves. We translocate, in other words, we move the transfer RNA from the A site to the P site. And then the whole process starts again. And you just keep adding amino acids in this way. And this is a very energy consuming process because every one of these things requires ATP. Termination happens when you reach a stop codon and the whole thing just falls apart. Okay? And we will pick up there tomorrow. Don't forget, Wednesday, your mini lab report is due. So don't wait until Wednesday to start working on it.